It's my pleasure to introduce the Cancer Biology program, which is led by Drs. Steffi Oesterreich and Ron Bukanovic, and Dr. Oesterreich will be giving the presentation. Come on up.
Good morning, everybody. It is my distinct pleasure to present the Cancer Biology program on behalf of Dr. Bukanovic and myself. Dr. Bukanovic is an expert in ovarian cancer cell biology and interaction with the tissue microenvironment. In addition to being the program co-leader for cancer biology, he's also the co-director of the Women's Cancer Research Center. I have been interested in endocrine treatment resistance in breast cancer and invasive lobular breast cancer, and just like Dr. Bukanovic, I'm the co-director of the Women's Cancer Research Center, in addition to being the program leader of cancer biology. We meet bi-weekly to promote intra- and interprogrammatic activities and collaborations. We mentor junior faculty, invite and host seminar speakers, and in addition to these tasks, we also have very unique responsibilities. For example, Dr. Bukanovic provides very crucial translational link to disease center leaders, and he directs the efforts to translate findings from CB members into clinical application, whereas I'm providing the link for the wet bench scientists to data science investigators. We have 12.2 million in cancer-related fundings. We have 43 members from 17 different departments. You can see our members are very collaborative. 15% of the more than 1,000 publications come from intra-programmatic interactions, 40% from inter, and 67% contain interinstitutional collaborations. This, as you heard before, we received a score of outstanding. There were concerns, they are listed here. Despite the attempts to focus the group, the aims of the group still appear somewhat broad. You already heard that we now split into two different programs, and within the cancer biology program, we have three specific focus areas: the hormone response and cell signaling, drug resistance and metastasis, and data science. There was little evidence of clinical trials generated by the group science. To address this, we recruited new faculty with a focus on physician scientists and translational biology, and there has been an increase in investigator-initiated clinical trials. There was a lack of multi-investigator trials. We were lucky to receive a U54 big data to better knowledge. This has recently ended, but the same investigators, mostly from our data science focus, are now participating in an Aurora study, which is BCRF funded, and it's a focus on matched primary and paired metastatic cancers, where the data is collected here and actually analyzed here. Dr. Monga recently received a P30, where one of the focus areas is liver cancer. Dr. Bukanovic is PI of a DoD Consortium Development Award, in addition to an OCRA program project grant, both focused on ovarian cancer. And finally, there have been resources from the program leadership to support breast and ovarian spore submissions. Specifically, we received team science pilot grant funding to foster these preparations. The aims of CB are listed here: to discover and elucidate cellular and molecular mechanisms of cancer initiation, progression, and metastasis, to transfer program discoveries to other cancer center programs to allow application in the clinic, and finally, to facilitate collaboration within and outside cancer biology, for example, through seminars, through regular meetings, focus symposia, as well as a yearly retreat. As I already mentioned, we have three focus areas: hormone response and cell signaling, drug resistance and metastasis, and data science. Within these three focus areas, we further focus on specific questions. For example, we try to identify critical signaling nodes, and we focus on estrogen and androgen signaling in the hormone response and cell signaling theme. We focus on tumor evolution and heterogeneity. And within the data science group, we specifically focus on integration of large omics data set with cell biology, as well as to translate complex cellular, tissue, and patient big data into opportunities for precision medicine. I'm now sharing 4 vignettes from you representing science from the 3 different focus areas. The first is highlighted, or is directed by Dr. Monga, an expert in liver cancer. Beta-catenin is frequently activated in liver cancer and causes druggable dependency. Specifically to test that, they made a mouse model which overexpresses activated beta-catenin and c-Met. These mice developed liver cancer, and through comprehensive molecular characterization, Dr. Menga and his team identified specific changes, and one of them, activation of glutamine-mTORC1 axis. They treated the mice with mTOR inhibitors and saw significant reduction of tumorigenesis. The second vignette comes from the prostate cancer group. Dr. Wang and Dr. Wip, his collaborator, have identified drugs that inhibit androgen receptor translocation from the cytoplasm to the nucleus. You can see this here. This is the chemical structures. These drugs are very effective in mouse models shown here, and the significant reduction of in vivo tumorigenesis is associated with an increase in apoptosis and a decrease in proliferation. These drugs have now been transferred to CT for further clinical development. The third vignette comes from the breast cancer group. A team science effort is ongoing to characterize invasive lobular breast cancer as the second most common histological subtype of breast cancer. The multi-investigator, multidisciplinary group have performed a very comprehensive characterization of the immune infiltration in lobular cancer. They have identified unique pathways and signaling for estrogen and growth factor receptor, and efforts are ongoing to characterize the unique metastatic setting in ILC. Concrete outcomes, for example, are formation of a Great Lakes ILC consortium, which includes, in addition to our center, Case Western and Cleveland Clinic, and OSU the James. In addition, a clinical trial, a window study, has been initiated, which is run through the TBCRC, but via the main site. I would like to mention that we are also very heavily interacting with patient advocate for this project. There's a National Lobular Breast Cancer Alliance group for which I'm chairing the scientific advisory board. And briefly, there will be the second International ILC Symposium next May in 2020, which we will host here in Pittsburgh. The last vignette comes from our data science group. Dr. Young and his colleagues use multidimensional big data to identify clinically relevant epigenetic changes. For example, he has used almost 10,000 samples from the TCGA data set to identify differentially methylated link RNAs. He identified EPIC1, which is hypomethylate, highly expressed in breast cancer, and is associated with worse outcome. The mechanism of action was identified. It directly binds to MYC and regulates MYC downstream target genes. More recently, he has used other data set where actually information is available for drug response. And through this, he identified link RNAs which are associated with drug response. For example, there's one link RNA shown here, which is associated with response to chemotherapy in patients with breast cancer. The highlights of CB include weekly seminars where we rotate between CB faculty, external speakers, as well as our own trainees. We have a cancer genomics special interest group, a translational research working group, and an annual retreat. On this slide, we summarize the value added. Of course, we tremendously benefit from the state-of-the-art shared resources. In addition, there is support for interprogrammatic seminars and symposia, for faculty recruitment, and a number of CB activities. We participate in interprogrammatic cancer center retreats. We have benefited from pilot funding for our members and obviously from the strategic planning discussions. We have contributed to the Cancer Center through expertise in basic cancer science, development of models, expertise in bioinformatics, and big data. We have transferred a number of agents to CT. Clinical trials are ongoing. We have organized interdisciplinary retreats. We have been very successful in mentoring of trainees and played a critical role in a number of T32 and team science efforts. We have addressed the catchment area through a lot of discussions with Dr. Rosenzweig and Dr. Robertson. These discussions are ongoing. For example, I meet with Dr. Rosenzweig every other week. At the end, we decided to focus on the 3 cancers which have the highest incidence and mortality in our catchment area, which is lung cancer, breast cancer, and prostate cancer. The lung cancer efforts are led by Dr. Stabil, who evaluates how tobacco impacts the tissue microenvironment. To promote lung cancer, she developed novel therapeutic and prevention strategies for individuals at high risk for developing lung cancer. A couple of examples for the breast cancer area. Dr. Rosenzweig and Dr. Lee study biology of racial disparities in breast cancer outcomes by adding biological variables, and Dr. Bravsky and Dr. Lee examined utilization of liquid biopsies, specifically circulating free DNA, comparing the academic and the community setting. The prostate cancer, I already mentioned the efforts by Dr. Wang and his colleagues to develop novel AR antagonists. This slide just shows a few examples for the great interprogrammatic collaborations. I don't want to go through this in detail. I already showed a slide earlier to really highlight the very extensive collaborations of our CB members. The future plans include to promote and foster team sites, for example, SPORE submissions, a PO1 submission, and continue our meetings we held on a regular basis. We would like further to promote mentoring and training efforts. We would like to increase cross-program collaborations, for example, through additional targeted mini-symposia. We have an effort ongoing using machine learning and systems biology to identify patterns of disease. Using the EMR and cancer registry, we would further like to enhance these efforts. And finally, use DMC as a platform for studies to understand unique biology of tumors and patients in our catchment area. And I'd like to thank you for your attention.
Dr. Pete and Paul.
Thank you for your presentation. A few questions. The first is, what do each of you uniquely bring to this program in terms of your role? You said you work together to do things, but what do you uniquely bring?
I'll start since Steffi's been talking a lot. And so my role is I am the translational link. So I'm a physician scientist, and one of the key roles I'm trying to play and playing is trying to identify both working with the basic scientist projects that we should be working on. So last week I met with one of the junior scientists, Dr. Osman Beo, and we were talking about how to make this more clinically relevant. So we met, some of it is starting at that very basic level, but then also when there is a project that is becoming clinical, how do we get that transited either to Dr. Chu or one of the other members in the group, Dr. Luke, who's now here, how do we transit that? So we have a new LOI that's just coming out of a project from one of the junior investigators here, looking at combining different projects for immunotherapy, and so that we're handing off to Dr. Luke. And so that's really the role that I'm playing as a critical link to the clinic.
Thanks. And maybe in addition to this, I can say that a specific role I play in CB is try to connect the investigators with our data science investigators. I think over the last few years, and maybe as a special result of working very closely with Dr. Lee, I started to work very closely with a number of data science investigators, and I'm really trying to get all the cancer biology investigators to initiate collaboration and continue collaboration and really make use of the expertise to combine wet and dry bench approaches.
And so you have as one of your themes data science. I would imagine data science is pretty robust across all programs in the Cancer Center. So why only in yours is this a theme, or should it, someone's going to ask, why is it a theme?
I think that data science for us, it's because it is mining the real basic biology. This is really the focus. It's not always, it's mining those basic biology, finding novel targets. And so, yes, will there be novel targets in immunotherapy? Yes, and are they using data science? But I think that broadly, when it was considered, that the data science really is identifying things like EPIC-1, these very basic mechanisms and models. And then ultimately, we can move that in the clinic, and that's one of the goals, but the idea is then to hand that off.
So of your 1,000 publications, the vast majority, 67%, are interinstitutional. Dr. Kasten made this comment earlier. Can you provide an example of a really impactful collaboration with another NCI center? Because so much of your portfolio is collaborating with other institutions.
Probably best for your
Yeah, I could mention one example. Is the work on invasive lobular breast cancer, where we interact with many other cancer centers. An example is that we played a crucial role in the TCGA analysis of ILC, which involves a number of cancer centers. In the end, it was published in a high-impact journal in Cell, and there were frequent in-person as well as conference calls to really attack that problem from a multidisciplinary angle.
And I have some other comments, but I'll wait for the other kind of site visit questions.
So I have a question. How much pilot project funding did you receive over the funding period?
We have received over the last 2 years, actually, well, less than 2 years, 14 pilot funds. And the total money,
It was about 750,000, if I recall.
Yeah.
that's correct.
Okay. So that should be mentioned with every program. I don't remember seeing it in the program before. Second question is, how are your priorities linked to the strategic plan priorities that the Cancer Center identified?
So I think that we meet as part of the REAC advisory committee, but we also meet regularly with Dr. Ferris. And so between the REAC and meeting with Dr. Ferris, kind of strategic planning is made. Some of that also comes with the community outreach to then feed back to us. So for example, a recruit that I'm working on, I would love to recruit somebody who's working more in the aging and cancer senescence and how that links, because we have a lot of strengths we develop in the breast and prostate, the early focus, which was chosen, but kind of thinking what's next. And so that's kind of an example of an interaction where we've worked with Dr. Ferris and the community outreach to, again, get that focus.
Okay. And
I think both of us are members of various strategic committees within the Cancer Center. One example, for example, is that I work very closely with Dr. Beckenest in the Training and Education Committee.
okay. And last question related to what Dr. Pete and Paul asked about. You're both in the women's cancer space. This is a broad cancer biology program. How do you make sure that you bring in all the other areas of cancer biology into the program?
Yeah, and so that's actually a very good question, thank you. So, but I think one of the great, actually, it's a strength that we both interact in the Women's Cancer Research Center, but also through the cancer biology program. And what it actually allows us to do is to bring in new contacts. And so by bringing in those contacts, it helps us define those intra-programmatic collaborations. For example, Carola Newman is in the Women's Cancer Research Center, but she's in the Genome Stability Program. But the work she's doing on chemosensitization for BRCA mutants is clearly very relevant to cancer biology. And so actually, by having the two, I actually think it's really a synergistic interaction between the programs.
Other questions?
I just had a few other comments. So I think based on, you hear us going back and forth about these specific roles, I actually really liked how Genome Stability said what you both they said what they functionally did in their program. And when you looked at your role descriptions on your slide, which is what the reviewers have, they're identical. That's why I asked that question right away. So really to describe what you individually bring to this program, and that's what Genome Stability did. And I thought it was very effective, and they were very different. And you can have as many leaders in a program as you want as a cancer center, but they have to be very articulately describe what the exact role is of each. The other thing is, I think this is a program, and to help kind of this catchments at the center, you know, it's a slide at the end, it's an afterthought almost because it's coming all the way at the end. Your program could actually put it at the beginning and set the stage for here's our catchment, here are the major cancers, breast, prostate's in their lung. And you're going to hear today about the research we're doing that's very responsive to the needs of the catchment, instead of kind of saying research that addresses the catchment tucked away at the back of the program. Because Stephanie, you said this really great thing, lobular cancer, second most common cancer, you're saying for breast cancer, but it'll be true for your catchment. That's really powerful. You know, I think flip all that around and say, you're going to hear work today. You're going to hear about prostate cancer and new drugs being made right out of this program in this center. So I think there's a much more provocative and kind of exciting way to present this. In this program, it would work, I think, to bring your catchment, how you're addressing the needs, instead of just, it's relevant, tucked at the end.
If I could riff off that just a little bit, all of the programs seem like there could be more of this. And so, you know, the COE, or maybe even the director's overview, should have a real clear statement about what our main issues are, which is kind of there, but it's a little bit fuzzy, and it's not entirely, you know, this is exactly what we're trying to address. And then each of the programs, I think, do have, but could really repeat that. Remember, our major focus of the catchment area in our cancer center is this, this, and this, and look what we're doing. Because you have such great examples, as do other people, but it's not coming across as like a unified mission across all the programs yet. And
Yeah, that's a great idea, thank you.
Yeah, this is the vision of the center. Here's the program's focus, and you're going to hear about it today.
so I won't have to repeat that in all the programs that are coming up, because that's what I was looking at. It's the same thing